To protect biodiversity, at least 30% of the world's land and sea areas should be effectively conserved and managed by 2030. The goal is clear, but where should those places be? To identify the most valuable regions, we should not only target hyper-diverse areas, but also consider where distinct biological communities occur. In other words, we should shift the focus from what to conserve to where else could this set of species be conserved. Moreover, evolutionary history should also be incorporated into estimates of biodiversity distinctiveness. By accounting for irreplaceability across both space and time, we will be aiming to safeguard global phylogenetic diversity. To do so, we will quantify and model phylogenetic uniqueness. Compositional uniqueness is the degree to which a community is different from all others. It is based on community dissimilarity metrics. If evolutionary history is incorporated, communities with species that are not closely related to other species are considered even more unique. Here, we introduce a novel framework to identify high-value biodiversity sites by focusing on which regions harbor unique sets of phylogenetic diversity and modeling the scale dependence of phylogenetic uniqueness at a global scale. Compositional uniqueness is typically measured as the average dissimilarity between a focal cell and all other cells. However, this central tendency approach overlooks the spatial structure of community dissimilarity and creates a scale dependence in average uniqueness measures. Phylogenetic endemism, a closely related metric, is similarly affected by such scale dependence. To avoid scale dependence and captured spatial structure, here we propose to model phylogenetic uniqueness analogously to distance decay patterns, the increase of phylogenetic uniqueness with spatial distance between a focal cell and all other. Note that, while distance decay models are built for the whole study area, distance increase models of phylogenetic uniqueness are built for each community. The parameters of this model inform about the spatial scaling of phylogenetic uniqueness of each focal community at a global scale. Thus, it is possible to differentiate global distinct but regionally less unique sites, evolutionary hills, from highly replaceable sites that are unique even at small scales, evolutionary islands. In other words, we can understand whether phylogenetic uniqueness arises from continuous diffraction of space and time or from abrupt spatio-temporal discontinuous. We have also assessed the efficiency of different biodiversity measures to maximize global phylogenetic diversity with the increase of protected area. Phylogenetic endemism was the most efficient criterion to preserve global phylogenetic diversity of vertebrates within the smallest land area. Notably, there is limited overlap with the current network of natural reserves. Moreover, when we applied our spatial scale framework to these cells, we observed that phylogenetic uniqueness may result from both evolutionary hills and evolutionary islands. This novel approach offers a valuable perspective for the implementation of the 30 by 30 target of the Montreal Global Biodiversity Framework, as it would allow preserving highly valuable sites with the minimum protected area.